This is going to be preparation for lecture 5. I'm going to spend time, 3 lectures, going through sort of translation and looking at 3 different sort of aspects, which is the first one is the genetic code, how it works, and some of the evidence that demonstrated the correct way it works. The next lecture will then be on how changes in the DNA leads to changes in protein function and therefore gives rise to a phenotype. And we'll revisit the sickle cell anemia example, and I may dig up a couple of other examples as well, if I have time. And then finally, the lecture 7 will just go over the steps, what actually happens during translation. And just like the other processes we've talked about, there's an initiation, there's an elongation, which is actually shown in this diagram that's on the front slide here, and then how the termination process occurs. And these are the learning outcomes, and I'll just, I will go over them briefly. Eventually, in a little bit, I will not have to go through them in detail. You can just read them. But I would like you to be able to, oh, there's a spelling mistake, be able to explain what we mean when we say, there's only one L, when we say that genes are collinear with their protein products. What does that mean? And that means also being able to explain the polarity. So both DNA and RNA, of course, and proteins have polarity, which means one end of the molecule is chemically different from another end of the molecule, and so therefore it participates in different kinds of chemical reactions. So anytime, think of north and south poles, you know, they're different from each other, and that's the same here. And there's a connection between the polarity of DNA and the polarity of amino acids. I would like you to be able to draw a generic amino acid. I'm not interested at all in you memorizing every single amino acid structure or, you know, any of that. There will be other courses probably where you might have to do that, but for me that's not important. But just the generic idea of how a peptide bond forms, because that is how translation occurs. It forms those bonds. Be able to describe sort of levels of protein structure, and that will help you in a later lecture be able to then see how making a change in the DNA changes the way proteins fold correctly, and then how that affects their function and leads to, of course, a phenotype. The experiments that show that the genetic code is a triplet and how we know it's a non-overlapping code. And then these all relate to, well, not this one, these two. These two relate to just how the genetic code was kind of unraveled. How was it interpreted? We learned that it was a triplet code. We learned it was non-overlapping, but then how did we figure out which nucleotide gives rise to which amino acids, right? And so there are a couple of sets of experiments that I'm going to go over in some detail, and you will have to do some calculations to demonstrate sort of that you understand how those experiments worked. And then finally, just be able to explain the bold terms in your own words. And when I say explain them, I also mean when you're giving an answer to a question that you use them correctly. So if you say transcription when you meant translation, that's going to be an incorrect answer. So we want the terms to be used correctly. It's very easy to make that mistake because they start with the same few letters, but you want to take special care, especially answering quiz or exam questions, because it will be marked wrong. Okay, so this is just a reminder of our sort of overall conceptual framework. We did the DNA replication part. We'll talk about meiosis and mitosis later. And then we did the translation part. And now this is where we are. And as I said, we're going to spend a few lectures here just going through some of the really basic material. Okay, so this is what you need to know about an amino acid. So the polarity of the amino acid, there is an amino group, an NH2 on one end, and a carboxyl group on the other, and this is referred to as the N terminus of the molecule, and this is called the C terminus. Um, and then the R is the part that's different for each amino acid. And all you need to remember is that this is the side chain group. This R group is what gives each amino acid its different kinds of properties, whether it's a polar molecule, whether it is hydrophilic, whether it's hydrophobic, whether it has a charge or is uncharged, or acidic or basic, any of those things. And those things dictate how a protein folds up and what it interacts with, and so on. And this just points out that at pH 7, the carboxyl group is missing a hydrogen and it's negatively charged, and the NH2 group has an extra hydrogen and is positively charged. So they're called ionized in that state. And I'm sure that you've covered this in other classes, but I want to make sure. And so these are the amino acids. They line up in the following way: the N terminus, and then the N terminus of the second amino acid interacts with the C terminus of the one before and makes a peptide bond. And this is the peptide bond. So that's the part you need to remember. I guess I didn't have to go up like that. And then the next amino acid is added with its N terminus binding to the C terminus of the previous one, and so on. And so that's how it works. And a water molecule is generated for each of these. So this is synthesis that produces, an H2O each time. And then here's our polypeptide with the N terminus and the C terminus. And I'm writing them in here because when there will be a place where I will probably give you a DNA, and you'll have to determine what the RNA is that matches that, and then you'll need to label the 5' and 3' of the, if they're not already done, of the DNA, and then of the RNA, and then you'll figure out what the amino acid sequence is, and part of your answer will be to indicate which is the N and which is the C terminus of your peptide chain. So just to be really clear about, what kind of things I would be testing you on, because those are the kind of things that I think at least show that you understand. So when translation occurs, it generates a long string of amino acids. And that's referred to as primary structure. And the protein is never just in this structure, because even while it is being produced during translation, even if only part of the protein has been produced, it will begin folding already. And the secondary protein structure, basically, depending on which amino acids, you can get these pleated sheets, beta pleats they're called. Or you can get helical shapes. And these form through hydrogen bonds between amino acids. So you've got an amino acid in one location and another in another location, and between them they can form a hydrogen bond, and that basically connects them and creates this secondary structure. But secondary structure isn't all there is either, because then these helical regions and the pleated sheets and so on will fold up into the tertiary protein structure. And so when there are interactions between these different regions, ionic bonds, hydrogen bonds, and other kinds of bonds, they fold the protein up in the correct structure. And then if a protein requires more than one amino acid chain, so think about hemoglobin, where 4 molecules are come together to form the hemoglobin inside a red blood cell, then that's called quaternary structure. Okay, so primary 1, 2, tertiary 3, quaternary 4. And in order for a protein to fulfill its function, it needs to be folded correctly. So if you had a protein and it was folded in a certain way, and there was a pocket, and that's where an enzymatic reaction took place. If you mess up which amino acids are in these various folded regions, you are going to disrupt the folding. And then maybe this protein, I can't possibly redraw it the same way, but maybe, the protein kind of folds around in some strange way so that now you don't have this little pocket that binds to a substrate and catalyzes a reaction. That means the protein isn't going to be able to do its job. Right? And sometimes you can make a mutation that just slightly alters the folding, so it has a very small effect on the protein, and it mostly works, but it's not quite perfect. And other times, even just one amino acid change messes up the protein function so that it can't do its job at all in the cell. So hopefully that's somewhat helpful. And so that's sort of basic protein structure. And when you think about the polarity of the protein, the N terminus and the C terminus, it turns out that it matches the 5' and 3' ends, respectively, of the coding strand of the DNA. So collinear refers to the fact that the polarity of the gene and then the polarity of the protein match each other, with the N terminus of the protein encoded by the most 5' of the nucleotides in the coding region, and the C terminus is encoded by the more 3'. And so the way this was determined was a number of mutations was identified, and in this case, this shows you the position of the amino acid. So this is amino acid number 15. It's supposed to be a lysine, but there's a stop codon, which we'll talk about later. So the protein, the mutation in the protein is up near the N terminus. Same with this at position 22. There's supposed to be a phenylalanine, but it has a mutation that changed the amino acid to a leucine, and that happens again up near the N terminus of the protein and up near the 5' end of the gene. You can make a connection between the position of an amino acid that is mutated in a protein and the position of the DNA where the change has taken place. There's a correspondence between the two. They line up with one another. And, this will seem probably obvious to you, especially if you already kind of know how this stuff works. But this was an important observation because it showed that the linear arrangement of the nucleotides was dictating the linear arrangement of the amino acids. So it was important work to understand at the beginning of trying to sort out, how genes actually function. So translation. Translation is the process. We've already talked about transcription. The mRNA sequence is made. It is processed if it's a eukaryote, and it's ready for now translation. And basically the string of amino acids that is put together, formed by making peptide bonds between them on the ribosome, is based on the sequence provided by the mRNA. So it's like the language of nucleotides is translated into another language, which is the language of amino acids. And so this is why I find this is a kind of cue that helps you understand what transcription and translation are. If you think about your transcript, which is a copy of your grades that you've earned in your classes, then you think of transcription as making a copy of DNA into mRNA. But the translation is when you take one kind of language and change it into another kind of language. So think of actually translating French into German or something like that. It's kind of the same idea. And as I said, I think in the last prep for lecture 4, that the actual translation is thought to, the ribosome contains both proteins and RNAs, and there is a fair bit of evidence that it's the actual RNA parts that are catalyzing the formation of the peptide bond, which is really interesting. So this is one kind of functional RNA. This RNA doesn't get translated into protein. It actually does a job in the cell. And same with transfer RNAs. These are RNAs that don't get translated into protein, but do a job in the process of making proteins. And so that's a little bit of an intro. What we're going to look at in this lecture, for the most part, is how the genetic code was figured out. How did they decode this and determine how it works? So I'm going to talk later in a few minutes about how it was known to be a triplet code. But when you look at the nucleotides of a messenger RNA, and you think about the process of reading them 3 at a time, because we know at this point that it's a 3 nucleotide code. So there's 2 different ways you could read it. You could read them in 3 sort of, and then 3, and then 3. So this is non-overlapping. So each unit of 3 nucleotides specifies a particular amino acid. But one of the, if you think about a gene and you think about being really super efficient, one possible way could have been reading it as an overlapping code. So the AUU would be the first codon, and then UUG would be the next one, and then UGC would be the next one, and then GCU, and so on. Okay, and so if you have some idea about how these things are read, and you need to know if it's an overlapping or a non-overlapping code, then how would you figure that out? And the way you would do it is you would make a mutation, a single mutation, and then you'd look to see what the consequence was to the protein. And although this would be easy enough to do these days, it was technically a very difficult approach to take. But if you had an overlapping code like this, then if you made a single mutation, suppose you changed this to an A, then the first codon would be changed, right? And then the second codon would be changed, and then the third codon would be changed. And so making a single mutation in the gene, the DNA, and just a single change of nucleotide in the RNA could make up to 3 amino acid differences. And so that's what you would predict. You'd predict, up to 3 different amino acid changes in the protein. Whereas if it's read in a non-overlapping way, you can only ever, if you have a single nucleotide change in the gene and then the RNA, it can only affect 1 codon. And by the early 1960s, they had basically done enough work to determine that the genetic code was a non-overlapping one. So if you had a mutated protein, it usually had only a single amino acid change, which is not what you would predict based on an overlapping code. And then just to get on to how we knew that the genetic code is a triplet code. And it's about making, and again, this was technically incredibly difficult work, so it took a long time to do. But basically, the idea was if you made a deletion of a single nucleotide, or 2 nucleotides, or 3 nucleotides, how did that affect the protein structure? And if you delete 1 nucleotide, for example, here, and here I'm just giving you sequence that is in little 3-letter words to kind of imitate the idea of the triplet code. And this is the sense. So this would be the equivalent of getting the correct amino acids, right? But if we delete 1 nucleotide, and so the first 2 words still make sense, and then none of these make any sense. And this is an analogy for all of these amino acids being the wrong amino acid. But I do think that using actual words can sometimes be easier to understand. And this is referred to as a frame shift mutation. And when this happens, at the site where the mutation happened, everything that comes after that is messed up. It's mostly the wrong amino acids. Every once in a while there might be a correct one just because of random effects, but basically you've messed up the protein. And it turns out that interestingly, if you have this mutant DNA, if there is an insertion mutation further on, you can kind of restore it. So what happens is the codons before where the deletion occurred make sense, and then there's some nonsense after the deletion. But then if you add another nucleotide, everything after that goes back to being read in these threes. And so this is called restoring the reading frame, because you took one away here, but you added one here. And so now almost all of it, this part doesn't make sense, the part in between the deletion and then the addition. So that part doesn't make sense, but the rest of the protein gives you the correct information. So it's the fat something or other that ate the big rat in this case. And of course, adding a nucleotide by itself, not in conjunction with the deletion, also gives us this. And we don't have to go through it in detail, but you can see perfectly well that all the words here are again, they're messed up. And so that's what a frameshift is. And we will be talking about frameshift mutations a little bit more in the next lecture when we look at actual DNA sequence and real amino acids. So this is kind of prep for that, as well as just understanding the genetic evidence. And so, like the first example on the previous slide, if you add two nucleotides, you still have this frame shift. So if you take away a nucleotide and then you take away another nucleotide, you've messed everything up. And if you add a nucleotide and add another nucleotide, you've got still this frame shift. So adding or removing two nucleotides doesn't fix the problem. However, if you have three additions or three deletions, you do get the reading frame restored. So if we remove the C and then remove the A and then remove the T, again there's a mess up between the first sort of removal and the final removal. So that part is messed up, but we still have the correct amino acid information to either side of that. And so this is the evidence that was used, that the code was read 3 bases at a time. So it's read, 3 nucleotides at a time. And that's the evidence. Now, this may be really clear to you, and you may know this already. And if not, if it's new and you find it a little bit confusing, just go through it yourself and try different additions and removals, and you'll see how this works. It just sometimes takes going through it more than once just to get the feel for it. Okay, so now they knew that the code was non-overlapping and that it was a 3 nucleotide code. And so then the idea was, okay, so which, if we read, a particular 3 nucleotides, what amino acid does that correspond to? And does it always correspond to the same one? Because if it doesn't, it's going to be really hard to figure out. And so Nuremberg is one of the first people that worked on this in his lab. And they used an enzyme called polynucleotide phosphorylase. And what they did was they took ribonucleotides, they put them in a test tube with the correct buffers and salts and all that sort of thing, and this enzyme. And this just basically strings together RNA molecules. So it just adds the nucleotides to each other and catalyzes that reaction without any kind of template. So they just incorporate whatever is in the solution at random. And of course, if there's only one kind of nucleotide, then what you're going to get is strings of uracil. And so they took that and made, first of all, these long RNA strands. And then they said, okay, let's just add ribosomes in the correct conditions and do an in vitro translation. When you work in vitro this way, you can get the ribosomes to attach to the molecule and translate without needing particular signals like start and stop codons, which we'll talk about later. yeah, in the next prep, most likely. And so when they took those poly-U, is what they're called, and had them translated, they made polyphenylalanine. So they had long strings of phenylalanine. So what that said was that UUU encodes phenylalanine. Okay, because we knew it was a triplet code, but we just didn't know which nucleotides coded for which amino acids. And then they do the same thing with poly A, poly G, and poly C, and they discover that 3 A's is read as lysine, 3 G's as glycine, and 3 C's as proline. So they know the first 4 codons. And now the next part involves mixing nucleotides in different proportions and so on. And I'm going to start a different prep for that because I really want, you should have a little break. You should think about all this stuff and then come to the next one ready to think through some calculations.